The Golgi apparatus. It is present in many eukaryotic cells. It is made up of membrane-bound sacs, also known as the Golgi body or Golgi complex. It is the only cell organelle to be named after a scientist, Camille Golgi. It is responsible for the modification, organization, and intracellular and extracellular transportation of macromolecules such as proteins and lipids. The structure of the Golgi apparatus consists of flat stacks of membrane-bound sacs called cisternae, many peripheral tubules and vesicles. The size of the Golgi apparatus varies, however, the organization is similar for any one kind of cell. It is bent and semicircular in shape. Some people have said it looks like a pancake shape, even a pita. The molecular composition and thickness on one end differs from the other. Golgi cisternae are an interconnected network of membranes. Normally, there are about 5 to 8 cisternae in one Golgi apparatus. However, as many as 60 have been observed by scientists. The cis phase is the receiving side. It receives molecules that have been synthesized in the endoplasmic reticulum via transition vesicles that float into the cytoplasm of the cell along the vesicular tubular network. The medial works in tandem with both the cis phase and the cis-trans phase to modify macromolecules. The trans phase is known as the shipping side. It plays a role in the modification of proteins. However, it also pinches off the modified proteins into secretory vesicles and gets them ready for transport to their final destination. Transition vesicles are membrane-bound sacs in which carry molecules from one place to another. They gather molecules by budding and forming clusters, then they fuse to a membrane-enclosed compartment for delivery. Secretory vesicles are the vesicles that are pinched off from the Golgi apparatus with the modified proteins packaged inside of them, ready for transport. Tubules are extensive networks within the Golgi apparatus. The function of the Golgi apparatus is complex and vital to the cell overall. The Golgi apparatus receives a large number of synthesized macromolecules from the endoplasmic reticulum via vesicles. These vesicles attach themselves to the cell membrane, and the contents then delivered to the lumen. The macromolecules are sorted, and the primary function of the Golgi apparatus is the modification of the macromolecules. Special enzymes within the different compartments of the cisternae allow for this modification. Some of the modifications include glycosylation by the adding of carbohydrates, some of the proteins, phosphorylation by the adding of phosphates to some of the proteins. It is also responsible for the synthesis of proteoglycans. With this, the Golgi apparatus takes a synthesized protein from the endoplasmic reticulum and attaches it to the glycosaminoglycan. It is the unbranched polysaccharide. The Golgi apparatus is also responsible for the sulfication of certain molecules. The process of sulfication takes place on the glycosaminoglycan attached to the proteoglycan, as well as the core protein. The molecules which gain the sulfur molecule from a donor, perhaps 3-phosphatoadenosine, 5-phosphosulfate, that need to go through the sulfication process enter via sulfotransferases into the lumen. The macromolecules that enter the Golgi apparatus from the ER come in as one thing, and after being modified can come out as something different, but always for what is needed. The proteins are labeled within the Golgi apparatus, thus determining their final destination. After the sorting and modifying, it is up to the transphase to package the modified proteins into secretory vesicles and set up for delivery to their final destination. These can be delivered either intracellularly or extracellularly. The Golgi apparatus also plays an important role in the transport of lipids around the cell, as well as the creation of lysosomes. The digestive enzymes remain in the cytoplasm until they are needed. Important molecules include nucleotide sugars that are imported to the Golgi apparatus from the cytosol. This is required by the Golgi in order to modify some molecules. The adenosine triphosphate is a required import into the Golgi apparatus as well for the process of phosphorylation of molecules. Glycolipids and sphingomyelin are synthesized within the Golgi. Oligosaccharides N-acetylglycosamine, and glucose. The Golgi apparatus plays a very important role within the cell. Without the Golgi apparatus, synthesized proteins would in a way be lost and stranded. They would not become what they are supposed to in order to benefit the cell. The Golgi apparatus is very important, especially when it comes to your DNA. Your DNA is copied by the RNA. So without the modification of the RNA, you would lose, well, you. Much research has been done on the Golgi apparatus. Research was conducted on how the proteins are actually transported within the Golgi network during the modification process. The CDC42 has been researched for polarity as it exists within the Golgi network. Also, research has been done on the role it may play in prostate cancer. So, if you want to be like something, be like the Golgi apparatus. Take what is given to you, make any necessary modifications, and then send it out into the world for the benefit of everyone.